Now for something completely different. How about an eye drop for the treatment of neovascular eye diseases, or even the prevention of neovascular eye diseases? We're here today with more than hypothesis and some confidence moving forward based on two important things. Number one is we've completed a phase 1/2 study in treatment-naive patients with wet AMD. 40 patients treated as monotherapy, eye drops only, for up to 8 weeks. 10 patients treated with a single injection of Lucentis, followed by eye drops for up to 3 months of treatment. On top of that, in parallel, we have done a significant amount of formulation work, because the one thing that we have noted as our dose-limiting toxicity in both animals and also now in our human clinical trial is that at higher doses we get an off-target effect on the cornea, corneal punctate keratopathy, that's reversible on discontinuation, but which really isn't satisfactory from a dose-ranging perspective on duration. We have now, however, a formulation that will reduce or even eliminate that potential risk moving forward. We've established regulatory requirements for how to develop this as a maintenance treatment, where we can actually engage patients and their caregivers in the chronic management of these diseases on an ongoing basis. We have intellectual property on our formulation that was just issued 3 weeks ago. We have our financing closed to enable an amended IND and CTA filings in Europe. And we will be clinic ready in the second quarter of next year with a significantly different formulation and dose range to work with. Why are we encouraged? Well, you'll hear this tomorrow. Dr. Scott Cousins is presenting our data for the first time at the late breaking session at 4:37. Don't come at 4:45, you'll miss it. And we'll have some more, but we have dose and time-dependent responses in patients at the 2 lowest doses, since our doses were actually were limited by the dose-limiting toxicity. So let me talk about that and let Scott do the work for tomorrow. This drug has always delivered high concentrations at the central retina and central choroid, almost independent of formulation. We can tweak it a little bit, but it always delivers the back of the eye because the magic is in the molecule itself. However, the cornea, for a very short period of time, an hour, maybe 2 hours, has significantly higher concentrations immediately after installation. And in some of these patients at certain doses, we see an off-target kinase inhibition at EGF, epidermal growth factor. Epidermal growth factor is important for corneal homeostasis, but we are not a very potent drug, so it takes basically 3 logs over what we would deliver for VEGF inhibition in order for that to happen. The key really observation here, thanks to my partner Marty Wax, is that we've noticed that in Tarceva-treated patients, we observed a corneal keratopathy that was identical to what we saw in our first in-man trial. So this is the key takeaway if a picture paints 1,000 words. Head-to-head versus our clinical formulation. The top formulation, the red dots, that is our clinical formulation. That is the corneal concentration in primates as a function of daily dose. And what you see is that as you increase dose, you get exponential increases in corneal concentrations. The bottom line with the blue diamonds is the current suspension formulation in primates. And what you see is that at doses from 1 to 10 mg per mil, you get a completely flat, low, and continuous concentration, well below the concentration required to inhibit EGFR. In fact, EGF typically inhibition starts at around 50 micromolar, so we are a log below that. So we have a very significant dose and safety range that we can work with going forward. What happens to the back of the eye concentrations? The bottom, the blue line is the corneal concentration, the top is the central choroid concentrations. We get very, very good dose-dependent concentrations in the central choroid at significantly higher dose range than we were able to dose previously. And that's why we're confident about going forward. In fact, we have not found a toxic dose on the cornea in primates at doses up to 20 times the human MTD. So, at this point, we expect to go into the clinic in the second quarter of next year with a dose that is 6 to 10 times higher than we were able to dose in the current study. And we are doing the chronic tox studies today to enable that CTA and IND filing in the first quarter of next year, with dosing to initiate in the second quarter. What we expect to do is something very similar to what we just completed, dose ranging monotherapy, but in this case for 3 months rather than just 8 weeks. as a way to pick doses for future development of a topical eye drop for the treatment and/or prevention of neovascular eye disease. This is our program as it is. The two key milestones that we have coming are presentation tomorrow of the clinical data, and then in December we will have the tox data in primates for enabling the CTAs and INDs. And what I would say is that you can imagine this enabling dose ranging for a phase 2 or even a phase 2/3 study, and depending on how aggressively you'd like to move, this could be an NDA in 2022 or 2025. We are looking for financing in advance of this trial, partnership even better, because we think that there's a benefit in the partner having visibility with us on the progress here so that this program can be prosecuted efficiently. And you have a chance here for payback and a significant value accretion in a space of about 12 to 18 months post-initiation of this next trial. These are the people that make the magic happen. I can't do it without them. I have a fantastic small team of 5 people that have done an awful lot of work and made an awful lot of progress. We have a great board of directors, and we're happy to have Third Rock, Novo, and SV Life Sciences continuing to support us going forward. Thank you very much.